Okay, so I'm recording on mine. I'll post it. So we're talking a little bit about cell signaling today. Kind of strange. That didn't work. Since something else is now working, we are good to go. All right, so talking a little about cell signaling, how cells communicate. This is kind of a short, brief chapter. However, this really kind of integrates with a lot of other chapters, a lot of other concepts, right? How cells communicate really is kind of a central concept, a central theme for all of biology. So we talked about cellular communication. There's just some key terms I'm going to give you at first. The first one's ligand. I think I've mentioned ligand before, but anything that binds to a receptor is known as a ligand. A kinase is an enzyme. It's an enzyme that phosphorylates things, right? That energizes things. So if you have an enzyme that's phosphorylating a protein, a motor protein possibly, that enzyme could be a kinase. GDP and GTP, right? They're just like ATP and ADP, right? GDP is guanine diphosphates. GTP is guanine triphosphates, the low energy, high energy forms. They're just another energy molecule other than ATP, just to be familiar with. Operates in very similar ways. We use mainly ATP, but GTP can work in some situations. Works in the same fashion as ATP. And the last one is a conversion of ATP to CAMP. CAMP stands for cyclic AMP. So adenosine monophosphate, meaning one phosphate. It loses two phosphates. And the cyclic is kind of referring to the shape. So ATP and CAMP. So these are key terms that we will use throughout today. Here's your outline. This is what we're going to focus on. We're not going to get through quite everything, but we will get through most of it. We're going to talk about local signaling, we're going to talk about long distance signaling, and then we're going to spend the majority of the time talking about signal transduction. Signal transduction is summed up very neatly as reception, transduction, response, and then we'll give you some specific examples of signal transduction pathways. Okay. So first, let's talk about local long-distance signaling. You already have a few examples of local signaling. Remember we talked about gap junctions in cells, right? Gap junctions allow neighboring cells to work in unison with one another. So whatever factors are controlling one cell can easily move through the gap junction into the cytoplasm of the adjacent cell. So that's kind of one type of communication. Could be hormones, could be signal molecules, could be inhibitor molecules, could be anything. The other one we should be familiar with is dealing with cell structure as well, is just cell-cell communication via receptors, right? We have glycolipids, we have glycoproteins. Those glycolipids, glycoproteins are identification cards for cells. So these are two types of cellular signaling you already kind of have been introduced to. Now, the other types of cellular signaling are broken down to local and then long distance. So when we talk about local, the first example is synaptic signaling, which occurs with what types of cells? Brain cells, neurons, right? Synapses. So we have synaptic signaling. And the other one is known as paracrine signaling. Very similar to synaptic, except it's just not neurons, it's normal cells. Just to give you some type of example here, right? Let's say you get a cut, right? So you get a cut, you cut your finger. The cells are damaged. Some cells were damaged, and some of the materials, the signaling molecules inside those cells are released to the surrounding environment. Those signal molecules could tell the surrounding cells, hey, it's time to start growing a little bit faster, increase the rate of cell division to try to repair the damaged area. Could be clotting factors, helping the scar to form, blood to clot, and so forth. So this is just signaling between cells that are close together, that are non-neurons. Here's our synapse, right? The impulse being sent down the neuron and then relaying the message to the next neuron. That happens with chemicals known as neurotransmitters, very good. Acetylcholine is a good example of that. So this is, again, a local type of signaling. The long distance signaling deals with the hormonal signaling. What system of the body does hormonal signaling fall into? What's that? The endocrine system, right? So this is part of our endocrine system. We really have two major control systems in our body. One is the one we always think about, which is the nervous system. The other one is the endocrine system, which controls the body via hormones. Great example would be like HGH, a hormone that's being secreted, that's targeting most of your body cells, especially your muscles and your bones, telling them to increase cell division, to increase growth, right? You're probably producing more HGH than I am at this point. I'm not really growing anymore. Could be epinephrine, right? The fight or flight response. Thank you. You're walking down the street, a big grizzly bear jumps in front of you, right? It happens all the time. You have to do one of two things. You're either gonna fight the bear or you're gonna run, right? How does your body get psyched up, ready to go? Part of it is the fight or flight response is the epinephrine release. Epinephrine is like adrenaline. You're gonna release adrenaline, which is gonna target the cells of your body. It's going to most likely increase respiration, increase heart rate. It's going to dilate your pupils. It's going to dilate your blood vessels, right? Because we're trying to get more oxygen, more glucose to your muscles, right? To power your muscles to either take the bear down. You know, I usually go with the headlock. But if the headlock's not going to work for you, then I would just say run.
Wait, what actually works is shoving your fist into the bear's mouth. Because they have really bad gag reflexes.
Okay.
So, like, they can't bite down if you, like, shove your fist down their throat.
I would make sure you vet that theory before you try it. I wouldn't just go off, like, reading on the internet and giving that a shot.
No, because there's a guy who did that. He saved himself from a bear attack.
That'd be an interesting experiment.
Yeah.
Just find a bunch of
Yeah.
bears.
I don't know if you get IRB approval, but maybe. All right. So there's kind of like the first types of signaling. The second one is some hormones are lipid-based hormones, which means they interact well with the cell membrane. A lipid-based hormone can pass directly through the cell membrane to cause change inside that cell. If it's HGH causing the cells to increase. Yes. Anyone, there's a few of them that are missing. None of them are the answers. There's a few of them like CO2. For some reason, a lot of the molecule terms are dropped out. Other types of hormones, protein-based hormones, right? Because they're hydrophilic, they don't pass through the membrane. So how do protein-based hormones relay their message into the cell? The answer would be, everybody? Signal transduction. To make this very, very simple, this is like the game of telephone. Right? Everyone's played telephone when you're little. Somebody makes up a little story. You tell it to the person next to you. They relay to the message to the person next to them, to the person next to them, to the person next to them. That's all signal transduction is. The people in signal transduction are proteins, though. One protein is being energized, phosphorylated, which is most likely going to phosphorylate another protein, and then cause some type of cellular response. There's some little details we'll deal with there, but that's basically it. So when we talk about signal transduction, we break it down into 3 steps. One is reception, that possible protein hormone binding to the receptor on the membrane. The second one would be transduction. It's still recording, wow. Would be transduction, which is the actual relaying of the message. This could be via proteins, could be via secondary messengers, which I'll mention in a second. And then you have response, which is a change in cellular response. Many times, that change in cellular response is going to be an increase or decrease in expression, gene expression. All right, remember, central dogma of molecular biology is simply DNA codes to RNA, which codes to proteins, right? Our DNA is just information to make proteins, enzymes, right? Signal molecules, right? A whole bunch of different proteins. So therefore, that cellular response could be, making proteins that are going to cause that cell to divide, speeding up cellular division. Maybe he's targeting to release insulin or some other type of hormone. So in any case, it causes a cellular response. So it is that simple. It's simply just reception, transduction, and response. Now, to add a little detail to it, we're going to talk about 2 types of signal transduction pathways. We're going to give you a little bit of detail about a G protein transduction pathway and then a tyrosine kinase transduction pathway. So you can kind of read through those. So here is our G protein signal transduction pathway. It's called a G protein because it has a protein that's energized not by ATP but by GTP, hence G protein. So here is our G protein coupled receptor. Notice there is no ligand bound to it. Right, therefore it is inactive, therefore our G protein is inactive. A signal molecule, possibly a hormone, binds to that G protein. Binds to that receptor. The G protein is attracted to the receptor, and it is then phosphorylated or energized with GTP, energizing that G protein. Now, the next thing that happens here is this. So 1, 2, 3 is it can start the pathway, activating other enzymes in the pathway. This enzyme here happens to be adenylyl cyclase. I always spell this wrong, so let's see if I get it right this time. Adenylyl cyclase, that is the enzyme there. Now, as I mentioned, some of these pathways can use just proteins in their pathway. So one protein will activate another protein and then another protein and just relay the message, kind of almost like a 1-for-1 going into the nucleus. However, some signals, some messages need to be amplified. Right? We don't want to be a 1-for-1. We want to increase this message exponentially. Now, it's hard to make a whole bunch of proteins because proteins are usually large, complex molecules. So what do we make instead? We make what we call secondary messengers. That can relay the message. And in this case, this adenylyl cyclase is taking a whole bunch of ATP and converting it into CAMP. But it's not doing a 1-for-1. In a very short period of time, it can make a large number of CAMPs. And that large number of CAMPs can amplify the signal going to the nucleus of the cell and increase the cellular response. So instead of just getting one message being relayed, by having the secondary messenger, we're now relaying 10, 20, 30 times the initial message, which is just amplifying that message.
But wouldn't 1 ATP just equal 3 CAMs?
What's that?
Wouldn't 1 ATP kind of equal 3 CAMs?
No, because you need the structural component of the molecule. Right? It's this whole large molecule with just 3 phosphate groups. It's not the individual phosphate groups that make up the CAMP. The rest of the molecule does. But very quickly, we can convert a whole bunch of ATPs into a whole bunch of CAMPs. That's the point of this process, is it's amplifying the message. So this is just the G protein coupled receptor. We also have a tyrosine kinase signal transduction pathway. Again, you just need to be familiar with the G protein, familiar with the tyrosine kinase. So when you see it, you know you're talking about a signal transduction pathway. That's what you need. You don't necessarily need the major details of each one. Being aware of secondary messengers is a good thing. So here, with the tyrosine kinase receptor, there's 2 of them. It's called a dimer because there are 2 receptors. Both receptors need to be bound to a ligand. When both receptors are bound, the ligand forms a dimer. The receptor forms a dimer, activating our tyrosine kinase receptor. Once that happens, the receptors start phosphorylating each other, adding phosphates to one, adding phosphates back the other way. The receptor becomes completely phosphorylated. Then the next proteins in the pathway start to be activated. We just activate a protein, which then activates another protein, and relays this message to the nucleus for a cellular response. These are really the 2 common pathways. They can be used to carry out a number of different functions, and different functions in different types of cells. So we have the tyrosine kinase, and we have the G protein. Now, watch a fun video. I posted a couple of videos to the AP Biology YouTube page.